I want you to do something for me right now. Just look at your hand, turn it over, look at the palm, look at your fingers. Or if you're driving, maybe just glance in the rearview mirror at your own eyes for a second.
Just a quick glance. Keep your eyes on the road.
Right. Keep driving safely. But what you are looking at is this incredibly complex biological machine. I mean, you've got a heart pumping liters of blood. You've got a brain processing this audio in real time, muscles keeping you upright.
It's wild when you actually stop to think about it.
It is, because you are basically a massive colony of roughly, 30 to 40 trillion individual units?
Yeah, trillion with a T.
Yeah.
It creates a bit of an existential crisis if you think about it too hard.
It really does. But here's the single most mind-blowing fact that I think biology has to offer. You, me, the listener, every single elephant, blue whale, giant redwood tree, we all started as exactly the same thing.
A single cell.
Just one single cell.
It is a profound realization. I mean, you go from one solitary microscopic cell to this complex, breathing, thinking organism. And that journey from 1 to a trillion, it isn't magic.
It's mechanics.
Exactly. It's pure mechanics. It's growth and reproduction on a cellular level.
And that is our mission today. We are doing a deep dive into those exact mechanics. We're tackling the cell cycle and cell division.
Which is really the bedrock of biology. If you don't understand how cells divide, you can't understand growth, you can't understand tissue repair, cancer, and you certainly can't understand why you look like your parents, but not exactly like your parents.
Right, exactly. Now, looking at our stack of notes and the source material we've gathered, we definitely have a specific target audience in mind for this dive. We know a lot of you out there are prepping for big exams, like the Neet, or maybe you're just a biology enthusiast who constantly gets tripped up on the jargon.
Oh, the jargon is brutal.
Because let's be honest, the terminology here is just a minefield.
It really is. You've got chromosomes, chromatin, chromatids, centromeres, centrosomes, biological nomenclature. Really does not do students any favors here.
It honestly feels like they named them all similarly just to mess with us.
It does. It feels almost intentionally confusing.
So our goal today is to cut right through that noise. We are going to clarify the notorious S-phase math, which is where everyone seems to lose points on exams.
The biggest stumbling block, yeah.
And we are going to break down the absolute beast that is meiosis PROPHASE.
Oh, yeah. The 5 substages.
The 5 substages, we are going to make them stick today. But let's start at the very beginning. The basic concept seems simple enough, right? You have a parent cell, it divides, you get 2 daughter cells.
Right.
Those daughters grow, they divide, and boom, you have a population.
That is the cycle. Growth and reproduction. But the key word there is cycle.
Cycle, right.
It implies a sequence. It's not just a random occurrence. Think of it this way. You can't just split a cell in half whenever you feel like it. You have to duplicate the instructions first.
Well, DNA.
Yes, the genome. If you divide before you've copied the manual, you're sending that new daughter cell out into the world with missing pages.
And that doesn't end well.
No, that cell dies.
Eesh.
Or worse, it malfunctions. So the cell cycle has to be this genetically controlled series of events. You duplicate the genome, you synthesize the cell constituents, the organelles, and then, and only then, do you divide into 2.
So it's a massive coordination game. Every step has to happen in a very specific order.
Exactly. And the stakes are incredibly high because you need those progeny cells to have completely intact genomes.
Okay, so let's talk about the timeline here, because I think this is the first place our intuition kind of gets it wrong.
Yeah.
I tend to imagine cells just constantly popping apart like popcorn. Just pop, pop, pop all the time.
Right, like a continuous boiling pot.
Yeah. But looking at the data from the sources, that is not really the case, is it?
Not at all. Let's take a human cell in culture as our standard model. The average duration of the human cell cycle is about 24 hours.
Okay, a full day.
Now, if I asked you how much of that 24 hours is spent actually splitting, like the physical act of division, what would you guess?
I mean, before reading this, I would have guessed maybe a third of the time. Like 8 hours, maybe 6.
1 hour.
Just 1 hour.
Approximately 1 hour. That is the M phase, or the mitosis phase. That's where all the drama happens. The other 23 hours, which is more than 95% of the total duration,
Right.
is spent in what we call interphase.
So if the cell cycle were a standard 24-hour clock, the actual division is just the time between 11:00 p.m. and midnight. The rest of the entire day is it just waiting around.
See, that is the historical trap. That's exactly what early scientists thought. They looked under these primitive microscopes, and during mitosis, they saw chromosomes moving, things dividing. It looked super active.
It looked like things were happening.
Exactly.
Yeah.
But during interphase, the cell just looked like a static blob. So they called it the resting phase.
Which I am assuming, based on our notes, is a terrible name.
It's a complete misnomer. The cell is metabolically incredibly active during this time. It is not resting at all. It's prepping.
Prepping.
Think of it like a chef arriving at a restaurant at, say, 10:00 a.m. for a dinner service that doesn't start until 5:00 p.m.
Oh, I like that.
The chopping, the making of the sauces, the organization of the stations that is interphase, and the actual dinner service where everyone is running around and it's total chaos, that's mitosis.
That makes perfect sense. You can't serve dinner if you haven't chopped the onions.
Exactly. Oh, and just as a quick side note to show how this varies, while human cells take 24 hours, yeast cells complete the entire cycle in about 90 minutes.
90 minutes. That's fast.
Very fast. So the timeline is specific to the organism, but the ratios generally hold up. Interphase takes up the vast majority of the time.
Okay, so let's walk through this prep work. Interphase is divided into 3 distinct parts. We've got G1, S, and G2. Let's start with G1.
So G1 stands for gap 1. It is the interval between the previous mitosis and the initiation of DNA replication.
But the word gap, again, implies a break or a pause.
Right. But it is strictly a gap in DNA replication, not a gap in activity. In G1, the cell is metabolically active and is growing continuously.
So it's bulking up.
Exactly. It's accumulating all the building blocks it needs, amino acids, nucleotides, it's producing enzymes, and crucially, it's checking its environment. It's asking, do I have enough nutrients? Is there enough space to divide?
It's a decision-making phase.
Yes. And if everything looks good, if it decides, yes, let's go, it moves into the S phase.
Which stands for synthesis.
Right.
Okay, stop everything. We need to slow down right here.
Ah, the danger zone.
This is the danger zone. This is the S-phase paradox that the sources emphasize. I was reading this, and this is where the logic gets really slippery for students, even for casual learners.
It's the most tested concept for a reason.
So in S-phase, we replicate the DNA. We synthesize a new copy.
Correct.
So the amount of DNA double.
Yes. You are synthesizing an exact copy of the genetic code.
But the source material emphasizes, like in bold letters, that the chromosome number remains constant.
That is absolutely correct.
That breaks my brain a little bit. If I have a suitcase and I double the contents of that suitcase, don't I need a second suitcase? How can I have double the DNA but the exact same number of chromosomes?
It's a fantastic question, and honestly, it's the #1 reason students fail questions on this topic. Let's use your suitcase analogy because it works perfectly here.
Okay, let's do it.
Imagine a single chromosome is a suitcase. Inside that suitcase, you have one set of clothes. That set of clothes is your DNA.
Got it. One suitcase, one wardrobe.
Now, in S phase, you manufacture a second, identical set of clothes. You copy the DNA. But instead of buying a brand new suitcase to put them in, you just stuff that second set of clothes into the exact same suitcase.
Okay, so now I have a really heavy suitcase.
Very heavy, stuffed to the brim. But if I am a baggage handler and I'm counting the luggage, I still only see one handle. I count it as one unit of luggage.
Yeah.
In biological terms, that handle on the suitcase is the centromere. As long as those 2 identical DNA strands, which we now call sister chromatids, are held together by that single centromere, they count as one single chromosome.
That clicks. That totally clicks. The chromosome count is defined by the number of centromeres, not by the sheer mass of the DNA inside it.
Precisely. You count the handles, not the clothes.
So let's do the math to make sure we have this absolutely locked in. Let's say a human cell starts in G1. It has 46 chromosomes.
Which you call diploid, or 2N.
Right, 2N. And let's say the actual physical amount of DNA is denoted as 2C.
Okay.
So it moves from G1 into the S phase. It duplicates all the DNA. At the very end of the S phase, what do we have?
You now have 4C amount of DNA. You have physically doubled the genetic material, but you still only have 46 chromosomes. You are still 2N.
Because we haven't split the handles yet.
Exactly. You have 46 double-stuffed chromosomes. The physical split doesn't happen until much later.
That is such a critical distinction. Listeners, if you are taking notes for NEET or just for your own sanity, write that down. S phase doubles the DNA content. It goes from 2C to 4C, but the chromosome count stays exactly the same. 2N stays 2N.
Yes. And there's one other small but really vital detail for animal cells happening during the S phase. While the nucleus is busy doubling all that DNA, the cytoplasm outside the nucleus is doing something too.
What's happening out there?
The centriole duplicates.
A centriole. That's the anchor for the ropes we'll need later on.
Exactly. We are going to need 2 anchors to pull the cell apart from opposite ends. That prep work happens right now in the cytoplasm during S phase.
Okay, so we've grown in G1, we've doubled our luggage and built our anchors in S phase. Now we hit G2, gap 2.
This is the final safety check. The cell continues to grow, and very specific proteins are synthesized here.
Like what kind of proteins?
Tubulin is a big one. It's the protein that actually makes up the spindle fibers in the ropes you just mentioned.
Ah,
It's the final countdown. Also, if any of the DNA was damaged during the replication process back in S phase, G2 is where the cell pauses and tries to fix those errors before it commits to division.
it's proofreading.
Exactly.
So that is the standard loop, G1, S, G2, and then into M phase. But there is an exit ramp, isn't there? I was reading in the notes about something called G0.
Yes, the quiescent stage, G0.
Quiescent. It sounds very peaceful, like a spa retreat for cells.
In a way, yeah. You see, not all cells need to divide constantly. Think about the neurons in your brain or the muscle cells in an adult heart.
Right, they aren't just churning out new heart cells every day.
No. Once they're fully formed, they're there to do a specific job, not to make copies of themselves. Adult heart cells don't appear to exhibit any division.
So what happens to them? Do they just get stuck in G1?
They exit the G1 phase entirely and enter this inactive stage called G0.
Now, when you say inactive, does that mean they go to sleep? Are they dormant?
No, and that is a huge trap for students. They are metabolically very active.
Hmm.
I mean, your heart cells are actively pumping, your neurons are constantly firing, they're working a very demanding 9-to-5 job. They just aren't on the reproduction treadmill anymore.
They are functionally active, but proliferatively inactive.
Exactly.
Can they ever come back from G0, or is it like a permanent retirement?
It really depends on the cell type. Some, like nerve cells, generally never come back. But others, like certain skin or tissue cells, sit in G0 but can be recalled back into the cell cycle if there's an injury.
Oh, interesting.
Yeah, if you cut your arm, the cells around the wound wake up, they leave G0, re-enter G1, and start dividing rapidly to patch the hole.
It's an on-demand system that is incredibly efficient.
Biology hates waste. Why spend the energy to divide if the organism doesn't need you to?
That makes total sense. All right, so let's say we didn't take the exit ramp. We've prepped in G1, we've double-stuffed the suitcases in S phase, we've proofread G2. Now we finally enter the main event, that 1 hour of pure drama.
M phase, mitosis, also known as equational division.
Why do they call it equational division?
Because the equation balances perfectly at the end. The parent cell has 46 chromosomes, and each daughter cell ends up with exactly 46 chromosomes. Nothing is lost, nothing is gained. It is a cloning process.
And we break this cloning process down into 4 distinct stages. The classic PMAT, prophase, metaphase, anaphase, telophase.
PMT, yep. Let's tackle them.
Let's do it. Prophase, the setup.
Right. So you have to remember, at the end of interphase, the DNA is loose. It looks like a giant bowl of microscopic spaghetti inside the nucleus. We call this loose form chromatin.
Okay, spaghetti.
Now, you can't move spaghetti easily. If you try to pull it apart, it tangles, it knots, it breaks.
So you need to pack it up tightly.
Exactly. Prophase is all about condensation. That loose chromatin starts to coil and wind around proteins, tightening up until it forms those classic, thick X-shaped structures we usually associate with chromosomes.
The suitcase is basically being zipped shut and strapped down.
And locked. And while the DNA is packing up, the nuclear envelope, the bag that holds the DNA, starts to break down and disintegrate.
It's getting out of the way.
Yes. Other organelles, like the Golgi apparatus, the endoplasmic reticulum, the nucleolus, they all seemingly disappear. The cell is clearing the decks for the main event.
What about the anchors we built? The centrioles.
Right. The centrosomes, which contain those centrioles, start moving to opposite sides of the cell. Think of them moving to the north and south poles.
Okay.
And as they move, they start shooting out these microtubules, creating what we call the mitotic apparatus. They form these star-like structures called asters. It looks a bit like a spider web forming across the entire cell.
Which perfectly sets us up for the next phase, metaphase.
Yes. The official start of metaphase is marked by the complete and total disappearance of the nuclear envelope. The chromosomes are now just floating free in the cytoplasm.
And isn't this the stage where we got those iconic pictures of chromosomes, the beauty shots?
Yes,
Yeah.
exactly. This is when the chromosomes are at their absolute most condensed state. If you ever look at a karyotype, you know those medical pictures where they line up all the chromosomes to check for abnormalities?
Yeah, I've seen those.
Those pictures are always taken during metaphase. Because that's when you can clearly see the 2 distinct sister chromatids held together by that single centromere.
And on those centromeres, there is a very specific little structure called the kinetochore, right?
Yes, kinetochores. Think of them as tiny disc-shaped trailer hitches attached to the centromeres.
Okay, trailer hitches.
Those spindle fibers, the spiderweb ropes coming from the north and south poles, they reach out and physically latch on to these kinetochores.
So it becomes a literal tug of war.
Exactly. The fibers from the North Pole grab one side of the chromosome, and the fibers from the South Pole grab the other side. And they pull and they tug back and forth until every single chromosome is lined up perfectly along the exact equator of the cell.
Forming what's called the metaphase plate.
You got it. And the cell actually pauses here for a moment. It has a checkpoint. It literally checks, is everyone lined up properly? Is every single kinetochore attached to a fiber?
Because I imagine if you split before everyone is attached,
then both sister chromatids might get pulled to one side. Someone gets left behind, and you end up with a massive genetic disaster. A cell with too many chromosomes and a cell with too few.
But assuming the check passes, assuming everyone is lined up on the equator, snap, we hit anaphase.
The split, the moment of truth.
What happens?
The centromeres physically split, the handle of the suitcase finally breaks in half, and suddenly, instantaneously, those two sister chromatids are no longer sisters.
Because they don't share a handle anymore.
Exactly. The moment they separate, they are officially considered independent daughter chromosomes.
Wow.
And as soon as they split, the spindle fibers start to shorten, rapidly dragging these new daughter chromosomes back toward the opposite poles.
The source material describes them looking very specific while they're being dragged, like V shapes or L shapes.
Yeah, because think about how they are attached at the kinetochore, which is on the centromere. So they are basically being dragged by their waists.
Oh, that paints a picture.
The centromere leads the way, pointing toward the pole, and the long arms of the DNA trail behind in the fluid. It's a very physical, violent, mechanical movement.
So they get dragged all the way to the poles, and then we hit telophase, which, from my understanding, is basically just hitting the undo button on everything that happened in prophase.
Pretty much, yeah. The chromosomes reach the poles, and they start to uncoil. They decondense back into that loose spaghetti chromatin mass. They lose their individuality.
The nuclear envelope comes back.
Right. A new nuclear envelope reforms around each of the two clusters of DNA. The nucleolus reappears, the Golgi and ER reform.
So at the end of telophase, you essentially have one large cell, but it has two completely distinct identical nuclei inside it.
Correct. Karyokinesis, the division of the nucleus, is complete.
But you're not done. You still have one big, unwieldy cell. You need to actually build a wall between them. And that brings us to cytokinesis.
Dividing the room, dividing the cytoplasm itself. And this is fascinating because it happens very, very differently depending on whether you are an animal cell or a plant cell.
Because plant cells have that rigid concrete wall around them.
Exactly. Let's look at animal cells first. The animal cell membrane is soft and flexible. So a ring of proteins basically forms around the middle equator of the cell and just cinches tight.
Like pulling a drawstring on a pair of sweatpants?
Exactly like that. It creates a furrow in the membrane that gets deeper and deeper until the cell just pinches entirely in two. Because it's moving from the outside edge toward the center, we call this centripetal division.
Outside in, centripetal.
Right. But as you said, plant cells have a rigid cell wall. You cannot pinch a concrete wall.
It won't budge.
No. So plant cells have to build a new wall from the inside out. They start sending all these little vesicles filled with building materials to the exact center of the cell. These vesicles fuse together to form a structure called a cell plate.
A cell plate.
And this plate grows outward, moving away from the center, which we call centrifugal growth, until it hits the existing side walls and fuses with them.
Inside out, centrifugal.
Yes. And that cell plate eventually matures into what's known as the middle lamella, which is basically the layer of biological cement that glues the two new adjacent plant cells together.
And during all of this, the other organelles, like the mitochondria and plactids, they just kind of get randomly distributed between the two halves, right?
Exactly. They get divvied up into the two new daughter cells.
Okay, before we move on, I have to ask about this specific detail mentioned in the notes. What happens if you mess this up? Like, what if you do the nuclear division, but you skip the cytokinesis part?
You create a syncytium.
A syncyt.
If karyokinesis is not followed by cytokinesis, you end up with a single cell that contains multiple nuclei. And if it keeps dividing its nucleus without dividing the cytoplasm, you can get a single cell with thousands of nuclei.
Which sounds vaguely terrifying, like a horror movie monster.
Well, you've probably eaten one
Yeah.
or drank one.
Really?
The liquid endosperm of a coconut, what we call coconut water, is essentially a giant syncytium. It is a massive cytoplasm soup filled with thousands of free-floating nuclei.
I am drinking a syncytium when I have coconut water.
Technically, yes.
That is wild. I will never look at a coconut the same way again.
Biology is weird.
Okay, so that is mitosis. It's clean, it's efficient, it makes identical copies, it keeps us growing, it heals our scraped knees. And this is a big, but that is not how we reproduce. That's not how we make babies.
No. If we used mitosis to make sperm and egg cells, we would run into a massive mathematical wall.
Right. It would be a terrible compounding interest problem.
Think about it. If a human sperm had 46 chromosomes and a human egg had 46 chromosomes, they fuse together, and the baby has 92 chromosomes.
And then that baby grows up, their gametes have 92, and the next generation has 184.
The genetic machinery would just break down. The species would collapse in a single generation.
So we need a totally different strategy, a reduction strategy.
Enter meiosis, also known as the reduction division.
Yep.
The fundamental goal here is relatively simple. Start with one diploid cell, 2N, and end up with 4 haploid cells, 1N. You have to cut the chromosome number exactly in half so that when fertilization happens, the diploid balance is perfectly restored.
It's kind of a paradox, isn't it? You have to aggressively reduce the number in order to conserve the specific species number across generations.
That's a perfect way to phrase it. But while the goal is simple, the process itself is not simple at all. Meiosis is often considered the beast of biology, specifically the very first stage, meiosis the cess.
Let's quickly clarify the overall structure first. Meiosis happens in 2 sequential rounds. You have meiosis the first, and then meiosis the second.
Right. And this is the crucial part that students miss. There's only 1 round of DNA replication.
1 round of replication, 2 rounds of division.
Yes. You copy the DNA once, but you chop the cell up twice. That is mechanically how you achieve the reduction.
Okay, let's face the beast. Meiosis the first, specifically prophase I. The outline calls this out as much longer and vastly more complex than mitotic prophase.
It is incredibly complex.
Yeah.
In fact, it's so complex that scientists had to break it down further into 5 distinct substages just to make sense of what's happening.
And these are the ones we promised to make stick today. Leptotene, zygotene, pachytene, diplotene, and diakinesis.
It sounds like you're casting a spell.
It really does. Let's take them one by one, starting with leptotene.
Okay, so lepto means thin and ten means thread. During leptotene, the chromosomes just start to become visible under a light microscope as these long, thin threads. They're beginning the compaction process. It's relatively straightforward.
Thin threads. Got it. Then we move into zygotene.
Zygo implies pairing. This is where the magic really starts. You see, in normal mitosis, chromosomes completely ignore each other. They just do their own individual thing. But in zygotene, the homologous chromosomes actively find each other.
Okay, define homologous chromosomes for us quickly.
They are the matching pairs. You have 23 chromosomes from your mom and 23 from your dad. Chromosome 1 from mom and chromosome 1 from dad are homologous. They code for the same general traits, even if they have different specific instructions.
Okay, so mom's chromosome 1 and dad's chromosome 1 find each other in the crowd.
Yes. They pair up side by side. This pairing process is called synapsis. And they don't just sit next to each other. They are literally zipped together by a complex protein ladder called the synaptomal complex.
It's a microscopic molecular zipper.
Exactly. And once they are zipped together, this paired structure is called a bivalent or a tetrad.
Why tetra? Where does the 4 come from?
Because remember, each of those 2 chromosomes already duplicated its DNA back in S phase. So mom's chromosome has 2 sister chromatids and dad's chromosome has 2 sister chromatids.
2 plus 2. 4 chromatids total in the zipped up package. A tetrad.
Exactly. Now, once they are fully zipped up, we move into the 3rd stage, pachytene.
And our notes highlight pachytene as the crucial stage.
This is arguably one of the most important events in all of biology. This is where you become unique.
Because of something called crossing over.
Yes. During pachytene, these recombination nodules appear on the chromosomes, and the mom chromosome and the dad chromosome, which are zipped tightly together, actually break physical pieces of their DNA arms off and swap them with each other.
They literally trade genes.
They trade entire sections of DNA between the non-sister chromatids. And it's not just random chaotic breakage. It's a highly controlled enzyme-mediated process.
What's the enzyme?
It's called recombinase. It precisely cuts the DNA strand of one chromosome and pastes it on the exact corresponding spot on the homologous partner.
So, just to make sure I grasp the magnitude of this, before this exact moment in pachytene, my chromosome 1 was entirely 100% inherited from my dad. But after this crossing over moment, it is mostly dad's DNA, but with a specific chunk of mom's DNA physically patched into it.
Yes. You have just created a brand new, genetically recombined chromosome that has never existed before in the entire history of the universe.
That is incredible. It is physical genetic engineering happening naturally at a microscopic level.
It is the ultimate remix, and it is the primary source of all genetic variation in sexually reproducing species. We'll talk more about why that matters later, but physically, that's what's happening.
Wow. Okay, so the swap is done. The genes are remixed. Now we move to stage 4, diplotin.
The breakup. The genetic swap is finished, so the synaptomal complex, the zipper, begins to dissolve. The homologous chromosomes try to separate from each other.
But the notes say they act sticky.
They do. They try to pull apart, but they remain physically attached at the specific points where that crossing over just happened. These X-shaped points of attachment are called chiasmata.
Chiasmata. It's
Yep.
like they're holding hands while they're trying to walk away.
It's a lingering goodbye, for sure. And here is a wild fact about diplotin. In the oogocytes, the egg cells of some vertebrates, including humans, this specific stage can last for months or even years.
Wait, years? A single phase of cell division lasts years.
Yes. A human female fetus actually starts the process of meiosis while she is still developing in her mother's womb. Her precursor egg cells go through leptotene, zygote, pachytene, they cross over, and then they enter diplotin, and they just hit pause.
They just stop.
They rest in diplotin. They wait there until she reaches puberty, which could be 12 or 13 years later, to finish the division process. Some of those eggs will wait 40 years before they resume.
Talk about a long pause. That is astonishing.
Biology has its own timeline. Finally, when the cell is ready to proceed, we enter the fifth and final substage, dikinesis.
What happens here?
The chiasmata, those X-shaped attachment points, they slide down to the very ends of the chromosomes. This is called terminalization. The chromosomes fully condense, the spindle fibers start to assemble, the nucleolus disappears, and the nuclear envelope completely breaks down.
Which means we are finally officially transitioning out of prophase I and into metaphase I.
We survive the beast.
That was a journey, just prophase I alone.
I promise you, the rest of meiosis goes much, much faster. But you have to understand the intricacies of prophase I to truly understand genetics.
All right, so metaphase I. We know from mitosis that metaphase is about lining up at the equator. How is metaphase I different?
In normal mitosis, individual chromosomes line up single file on the equator. But in metaphase I, the homologous pairs, the bivalents, line up together.
Oh, they line up 2 by 2.
Right. Mom's chromosome 1 and dad's chromosome 1 are side by side at the equator. The spindle fiber from the North Pole grabs mom's chromosome, and the fiber from the South Pole grabs dad's.
Which sets up a very different kind of tug-of-war for anaphase I.
Exactly. This is the second huge trap for students. In mitotic anaphase, we split the centromere. The sister chromatids get torn apart. But in anaphase I of meiosis, we do not split the centromere.
Wait, so the suitcase stays closed?
The suitcase stays firmly closed. The homologous pairs separate from each other. Mom's whole suitcase gets dragged to the left pole, and dad's whole suitcase gets dragged to the right pole. But the sister chromatids inside those suitcases remain attached to each other.
So you are separating the homologous sets. You aren't tearing the individual duplicated copies apart yet.
Correct. And this right here is the actual reduction step.
How so?
Well, think about the numbers. You had 46 chromosomes lined up in pairs. 23 pairs. When they get pulled apart, 23 whole chromosomes go to one pole and 23 go to the other. You have physically reduced the chromosome count at each pole from 46 to 23.
Even though those 23 chromosomes still have double the DNA because they still have 2 chromatids.
Yes, they are still double stuffed, but the count has dropped to haploid.
Okay, that makes sense. Then we hit telophase I, the nuclear membrane briefly reappears, cytokinesis splits the cell, and now we have 2 distinct cells.
2 haploid cells.
Do they get to rest now?
They get a very short break called interkinesis.
But no DNA replication, right?
Absolutely no DNA replication. If we doubled up again, it would defeat the whole purpose of the reduction we just did. So after a quick pause, they dive straight into meiosis II.
Which our outline affectionately calls the mitosis mimic.
Because mechanically speaking, meiosis II is virtually identical to normal mitosis.
So prophase II, the chromosomes condense again.
Yeah. Metaphase II, the chromosomes line up single file at the equator, just like in mitosis.
And anaphase II.
Now, finally, we split the centromere. The handles break. The sister chromatids are torn apart and dragged to opposite poles.
Just like normal mitosis.
Exactly. And telophase II wraps it up. The nuclear envelopes enclose the groups of single chromatids. Cytokinesis divides the cytoplasm again.
And the final result.
You started with one single diploid cell at the very beginning. You went through one round of replication, 2 rounds of division, and you end up with 4 haploid daughter cells.
And because of that crossing over back in Pachytene, every single one of those 4 cells is genetically unique.
Completely unique.
Yeah.
None of them are identical to each other, and none of them are identical to the original parent cell.
It is an incredibly elegant system. Complex, but elegant.
Very much so.
So let's zoom out for a second. We've crunched all the mechanics. We've done the suitcase math. We survived the beast of PROPHASE. Why does all this actually matter? What is the big so what for a student or just a curious human?
Well, let's look at mitosis first. Mitosis is fundamentally about survival and maintenance. It is how you grew from a single fertilized egg into a trillion-celled adult. It's how you replace dead skin cells, how you heal the lining of your gut, how you make new blood cells.
Without mitosis, we are just static and eventually dead.
Exactly. It also solves a very practical geometric problem for the cell, which is maintaining the nucleocytoplasmic ratio.
What does that mean?
As a cell grows, its cytoplasm gets larger and larger, but the nucleus, the control center, stays the same size. Eventually, the factory floor gets too big for the manager in the control center to oversee effectively. Division restores that balance. It keeps cells at an efficient, manageable size.
That makes total sense. And the outline also mentions plants specifically regarding mitosis.
Oh, yes. Plant growth is fascinating. Unlike animals, which generally stop growing at a certain age, plants grow continuously throughout their entire lifespan.
Right. A tree just keeps getting taller and wider.
And that's because plants have specific meristematic tissues, like the apical meristome at the tips of roots and shoots, and the lateral cambium in the trunk. These tissues are just engines of meiosis, constantly dividing, allowing the plant to grow forever.
Okay, so mitosis is growth, repair, and maintenance. What about meiosis? Why do we care so much about this complicated reduction division?
Meiosis really does 2 critical things for life on Earth. First, as we discussed, it conserves the specific chromosome number across generations. It ensures that humans with 46 chromosomes, dogs with 78, and wheat stays wheat.
If the chromosome numbers drifted every time we reproduced, species stability would just collapse entirely.
Exactly. But the second thing it does is arguably even more important, variation.
Remix.
Yes, that moment in Pachytene, that shuffling of the genetic deck. That variation is the raw material, the fundamental fuel for the entire process of evolution.
Because if we all just cloned ourselves via mitosis, we'd all be exactly the same.
And if we were all exactly the same, one highly robust virus or one sudden shift in the climate could wipe out the entire human species. We would all have the exact same immune system blind spots. We would all have the exact same vulnerabilities.
So variation is resilience.
Variation is survival. It gives nature a diverse menu of traits to select from when the environment changes. It is the reason why some people survive plagues or can digest certain foods or adapt to high altitudes. It's why you are uniquely you.
It is a beautiful, necessary balance. We have mitosis for stability and conservation within our own bodies, but we rely on meiosis for uniqueness and adaptability as a species.
That is the poetry of biology right there.
All right, before we close out, let's quickly recap the absolute key takeaways for our listeners, especially those heading into an exam room soon.
Number 1, do not underestimate interphase. It takes up 95% of the cell cycle. It's not a resting phase. It is a highly active preparation phase.
Number 2, the S-phase paradox.
Yes. Remember the suitcases. DNA content doubles 2C becomes 4C. But the chromosome number stays exactly the same. 2 stays 2. Double the luggage, same number of handles.
Number 3, the difference in the split during anaphase.
In normal mitosis, and in meiosis the second, you split the centromere. The sister chromatid separate, but in meiosis the first, the homologous pairs separate, and the sister chromatids stay glued together.
And finally, meiosis prophasis spall. Remember the spell. Leptotene, zygotene, pachytene, diplotene, dikinesis.
And specifically, remember that pachytene is where the crossing over actually happens. That is the high-yield question on almost every exam. Pachytene equals crossing over.
You know, I actually want to leave the listeners with a final thought specifically on that point. We talked about that enzyme earlier, recombinase.
Oh, arguably my favorite enzyme.
It really is fascinating to think about. There was a specific physical moment, a tiny microscopic event inside your parents' biology, where DNA was literally being cut and pasted by that enzyme.
A literal molecular scissors and glue.
Exactly. And the specific, totally random combination of traits that resulted from that swapped the traits that make you, you. Your laugh, your eye color, your height, even weird things like your predisposition to thinking cilantro tastes like soap.
Yep, all determined by the shuffle.
All of it was physically engineered during that tiny, fleeting window of time in PROPHASE.
It's staggering. Without that specific enzyme, recombinase, and without that specific reshuffling process, the immense diversity of life as we know it simply wouldn't exist. We would be living on a much, much duller planet.
So the next time you look in the mirror, maybe not the rear view mirror this time, a real mirror, just remember, you are the result of millions of perfected cell cycles and one very specific, very lucky shuffle of the genetic deck.
I really couldn't have said it better myself. It's a masterpiece of engineering.
Well, thank you all for diving deep with us today into the mechanics of life. Good luck with the studies. Keep reviewing those phases and keep marveling at the machinery inside you.
See you next time.